smart, and his depth of biological knowledge somehow never ceases to amaze me. He's also very fearless. I often joke in the lab that when there is a paper that gets published on some new neuroscience tool, he'll find a way to get his hands on it before the paper is even fully published. And oftentimes it's some type of wild virus, which keeps our safety department on its toes. Julian has a great sense of humor. He's an awesome colleague in the lab. Beyond the work that he'll tell you today about, he's been on an author in 2 of the papers that have recently come out from the lab, helping others, in particular with biochemical approaches. Julian is incredibly independent and creative. I remember the first time, or one of the first times we met, sitting in front of the board and thinking about, what projects you might want to work on, and he's interested in studying metabolism. And I think to myself, I know nothing about metabolism. How are we going to do this? And of course, like, he just gets going on this project and takes off and conceives the ideas and figures out how to do the experiments, analyzes the data. And I'm really proud of the story that has come out of this that we're here today about. Julian, I think you have an incredible, future ahead of you. And I hope that over the last few years, you've learned from me as much as I've learned from you. We'll miss you. But for now, join me in welcoming Julian.
Right. Thank you so much for that wonderful introduction, Sanisha. Can everyone hear me? Okay, great. So yeah, I'm pretty excited to talk to you guys about the work that I've been doing in Sanisha's lab, mainly regarding this project that I'm working on right now, which involves a preoptic neurocircuit that regulates blood glucose homeostasis. And so, kind of to get into it, when we think about the stuff that we're studying in the Hervatine lab, what we're really looking at are 2 things. Thermoregulation and metabolism are kind of our bread and butter. And so I think it's important to ground ourselves in the way in which animals thermoregulate. And canonically, these are one of 2 ways. So we have ectothermic animals, which represent the majority of all animal species, actually, such as snakes, reptiles, lizards, amphibians. And these are animals that keep their body temperature basically in line with what the ambient temperature is. Now, the other option is endothermic animals. And those are basically mammals and birds. And these are animals that will defend their body temperature to generally around 37 degrees Celsius. And there are advantages to each of these options. But really, what kind of links the 2 of them is this process of metabolic control of body temperature. And so in the context of an ectotherm, right, this is not dramatically difficult, right? Your metabolism changes in line with what the ambient temperature is. But for the endotherms, there's actually a tremendous energetic cost to maintaining your body temperature. We devote a significant amount of our overall metabolic rate to that, to the point that when we're at ambient temperatures that are close to 4 degrees Celsius, we're actually maxing out the metabolic rate. And so these 2 things are incredibly intertwined with one another, to the point where roughly 70% of overall metabolism is accounted for by thermogenesis when we put an animal at 4 degrees Celsius. So what we really study, though, is parts of biology in which there are deviations from these normal thermoregulatory behaviors. And 2 of the most major ones that we see are torpor and hibernation. Now, when we think about hibernation, you might think of, say, a brown bear in a den or a hibernating hamster or something like that. And this canonical sort of behavior in which over a 3-month period or so throughout the winter, these animals will go into 2-week bouts where their body temperature drops to 4 degrees, and they're basically in a stasis, pausing only to arouse basically once a day, once every 2 weeks. Now, beyond this, we have to understand that these behaviors are actually pretty well conserved amidst the mammalian clades. There are 2 separate behaviors that we're talking about here. We're talking about daily torpor and hibernation. And whereas hibernation is more of a long-term behavior in which animals are actually able to sustain these large periods of deviation from normal body temperature, daily torpor is more of a survival strategy. So when animals are food deprived, they will enter into this state of daily torpor where, again, they drop their body temperature to near ambient levels. And luckily for us, it just so happens that our favorite model organism, the mouse, Mus musculus, is an animal which is able to enter into torpor. And this is a great, this makes a great tool for us. This makes a a great tool for us to actually study this behavior and understand what these animals are doing in this state. And I think it's good to pause and kind of think about how this might actually be applied, how results from these types of experiments might be applied to things within human health. And so sort of the two pie in the sky things we can think about are space travel, where say you have humans traveling across light year distances in which it would be more beneficial if you could prevent the aging of these organisms, or you could reduce their metabolic rate and reduce their need for food consumption so that you can make these long travels. And so if we can understand how we can induce these states of hibernation, these states of stasis, we can actually make it such that we could put humans into these states. The other one, which is a little less pie in the sky and probably more reasonable for us to get within the next couple of years, is this idea that during hibernation, animals, despite long periods of inactivity, don't display muscle wasting that you would expect to see in, say, bedridden patients or people who also have similar amounts of inactivity. And so if we can understand the mechanisms by which hibernators are able to prevent these sort of inactivity-based atrophy from occurring, we can apply that to better human health. So now that we've sort of hinted at this idea of, what is torpor and what are we studying here, how do we actually look at this in the lab? So fasted mice enter into torpor, and the way in which we actually track this is pretty simple. We take telemetric temperature probes and we implant them into these animals. And then we take those animals and we put them on temperature platforms, and we can monitor their body temperature over the course of a 24-hour period. So in a fed animal, what this might look like is you have normal oscillations of body temperature around 37 degrees Celsius, but by and large, we're maintaining an elevated body temperature. However, when we fast these animals, what we observe is this stereotyped pattern of body temperature depression that occurs largely between the transition between the dark and the light sun. And what we see is these periods, it could be one, it could be two, it could be many. But we see these periods of body temperature depression that are near ambient, so around 24 to 25 degrees Celsius, that persist for about 4 to 5 hours. And this is what we would call torpor. Now, torpor is not only a hypothermic state, but also, if we look at the metabolic rate of the animal as read out by the oxygen consumption, we can actually observe that it's also a hypometabolic state. And we can see that during torpor, these animals actually have roughly a 60% reduction in their metabolic rate. And so we can define torpor as both a hypometabolic and a hypothermic state. And this is something, a through line, that I want you guys to keep thinking about throughout the duration of this presentation. So now that I've described torpor, why don't we take a look at what it actually looks like? So if we look at this animal right here that's had food removed from the cage, it will continue to search for food over the duration of the fast. But eventually, it will stop searching and adopt this posture right here that we're seeing and drop its tiny fish. And this is what we would call torpor. Now, the animal's not sleeping. As you can see, it's very easily aroused by just simple stimuli. And in fact, this is what makes studying this state so hard, the fact that it is very reversible and very transient. So now that we understand what torpor looks like, we can kind of think about it in the context of recent work that's been done by Sinisha. And so he asked the simple question, what are the neurons that control torpor? And you might ask yourself, well, why do we think that neurons are actually controlling torpor at all? Well, it's been known for a long time that the preoptic area is the thermoregulatory center of the body. So that's how endotherms maintain their body temperature at a certain level. And through a series of experiments in which Sinisha used activity-based tagging of certain neurons, he was able to identify a structure within the brain called the AVPO, or anteroventral preoptic area, in which a population of glutamatergic PACOP expressing neurons was able to be identified as associated with torpor activity. And moreover, when these neurons were stimulated synthetically, he was able to show that he could recapitulate the body temperature drops observed during natural torpor. So now I'm going to align a lot of work that Sinisha did over his postdoc, but he was basically able to show that via fiber photometry, these neurons are active during torpor. He was able to synthetically induce the torpor behavior via activation of these neurons, demonstrating their sufficiency. And then he was able to show their necessity by inhibiting the activity of these neurons and showing that you no longer get the characteristic decreases in body temperature. And so this sort of set the stage for a lot of the work that I did. But before we get into that, I want to talk about how do we as scientists manipulate the brain? How do we do neuroscience in order to understand these things? And so the main tools that we use are AAV-mediated gene delivery, so adeno-associated virus-mediated gene delivery, where we produce genetic constructs that we put into viruses that we then transduce into neurons. And we can actually selectively determine where these constructs get expressed using what's known as the Cree-Lock system. So the details of the Cree-Lock system are not terribly important, but in brief, what you need to understand is we can use Cree-dependent vectors in which we flip the orientation of the gene. Now, when the DNA processing machinery reads this, it's not able to produce a functional gene product, and so we don't get a protein. We get no activity of that on whatever construct we put into those viruses. But in the presence of Cree, it flips the gene into the right orientation, and we get a gene product, and we're able to manipulate the neurons as we would like to. Now, we do this by exploiting transgenic animals. So in this case, we express Cree in the VGLUT2 population neurons using these VGLUT2 high-res Cree animals. And this allows us to genetically segregate activity of particular viruses to these neurons specifically. So in practice, what this looks like is we will load a Cree-dependent AAV into needles. We'll perform stereotaxic surgery where we anatomically target a particular region of the brain, in this case, the AVPOA. And then, using Cree-restricted expression, we can actually get expression of our construct of interest in the particular area that we want. And so, in doing this, we can then manipulate neurons. And the way to manipulate these neurons is via chemogenetics and optogenetics. So the nuances of these are not super important to know, but what's worth knowing is that our chemogenetic approaches allow us to transduce these neurons with a protein known as a GQ DREAD. So this is a designer receptor which binds a synthetic ligand, which is inert in all neurons that don't express our gene of interest. This is known as CNO or clozapine N oxide. And upon binding the clozapine N oxide, these GQ DREADs depolarize the neurons, which leads to their activation. So it's an activating construct. Similarly, we do Cree-dependent optogenetics, in which we transduce these neurons with channel rhodopsins, which operate by a different mechanism, but in response to blue light, they increase the excitability of the cell and activate it. And so using stereotaxic administration of these viruses to different regions in the POA, we're able to selectively control the activity of these neurons. So now that we have that out of the way, I can really begin to address the question that I wanted to ask, which is, we know a lot about the thermoregulatory properties of these neurons. We need to characterize them very well. But what we don't understand is how do these neurons actually induce the systemic changes in metabolism that we see? Remember, torpor is a hypometabolic state, and we don't really understand the actual mechanisms by which that state is induced. So to begin to probe this question, what we did was we took animals and we placed them into metabolic cages. And these metabolic cages allow us to measure temperature, activity, gas exchange, and other things. And we transduced these animals with the GQ DREAD. And upon stimulating these animals with CNO, we were able to show that we could recapitulate not only the body temperature decreases that we observed during natural torpor, but also the decreased metabolic rate that we observed. So using the system, we've already phenocopied the two experimental or the two behavioral characteristics of torpor that we expect to see: hypometabolism and hypothermia. But given that we're using these metabolic cages, we're actually able to obtain more information than just this. And we can actually develop a derived parameter known as respiratory exchange ratio, or RER. Now, what RER is canonically within the physiological sense is it's a proxy for fuel usage. So the numbers will span between 0.7 and 1.0. And if you take on a value of 0.7, this corresponds to entirely fatty acid oxidation. A value of 1.0 corresponds to carbohydrate oxidation. And anything in between indicates mixed metabolism. And so interestingly, what we were able to observe is that when we stimulate these neurons in fed mice, we're able to see a shift from mixed metabolism to entirely fatty acid oxidation, suggesting that we're shifting away from a carbohydrate or glucose-based metabolism towards a fatty acid-based oxidative paradigm. Now, this was interesting to us, but we had to prove one thing before we really wanted to tackle this question. And plain and simple, that was, well, is this all just an effect of temperature? you might imagine that you could observe this effect simply as a result of the animal's body temperature decreasing, which has an overall effect on the enzymatic machinery of the animal and would lead to the result that we observed. And so to test this, what we did was we elevated the ambient temperature of the room that these animals were housed in from 24 degrees to 32 degrees. And when we stimulated these animals, what we observed is that their body temperature only decreased to 35 to 36 degrees, as shown in that blue line right there. This is well within the normothermic range. And so we could imagine that this would allow us to isolate the effects of temperature from just the activity of these neurons. And what we found was, when we stimulated these neurons, even at 32 degrees, where the animal's body temperature was not substantially reduced, we still observed the RER effect that we saw in the original experiments. And this is quantified right here. And so, because we see no difference, regardless of the ambient temperature, regardless of the core temperature changes of the animal, we conclude that this is not a temperature-dependent effect, and that it is solely dependent on the activity of these neurons. And so, to continue with this, that led us to three main questions. One, can we determine what metabolic changes are controlled by these neurons? Two, can we dissect how these neurons signal metabolic changes to the periphery? And three, can we then assess the function of these neurons in normal physiology? And so to address these questions, what I first wanted to do was answer, what are the metabolic changes that are happening? We know that we can activate these neurons. We know we can induce this state of pseudo-torpor or torpor-like state, but what is the actual metabolic consequence of turning on an AVPOA neuron? And so to ask this question, we performed an unbiased experiment wherein we took animals and transfused them with our GQ dreads or a control construct. We then took blood samples at baseline and injected them with CNO, and then took regular time points of blood samples afterwards and processed them for serum metabolomics, in which we could assess how the metabolome of these animals changed under GQ stimulation or under just normal controlled circumstances. And so looking at the metabolome of these animals, after 2 hours of stimulation, comparing GQs to controls, we observed three main things. If you look at the left side of this volcano plot, what you'll notice is that basically every glycolytic or TCA cycle metabolite is represented there as being downregulated. And if you're familiar with basic biochemistry, you know that this is the main way in which glucose is processed. These are the main catabolic machineries for glucose in the body. And so this is consistent with a hypothesis in which glucose metabolism is decreased. Now, moreover, on the right side of this, we see that one of the main ones that is actually increased is glucose. And this is again consistent with the idea that glucose is not being uptaken from the plasma and is continuing to circulate in the blood. And finally, we see that ketone bodies are relevant. So 3-hydroxybutyrate is a ketone body, which is a product of fatty acid oxidation. And so we know now that by stimulating these neurons, we're actually inducing a fatty acid oxidative paradigm simply via the turning on of these neurons. So this led us to want to ask, okay, are we really shifting away from glucose metabolism? And to answer this question, we performed a very simple physiological test known as a glucose tolerance test. Now, I'll take you guys through this because this is a very core experiment for a lot of the work that we're doing. But what we're seeing here is we fast the animals for 3 hours. We take a baseline blood glucose measure. We then inject the animals with CNO, and we allow them to go into torpor for 1 hour. After this, we take a baseline blood glucose sample, and we inject them with a bolus of glucose. And after this, at regular intervals, we then sample the blood glucose levels and see how they renormalize. So in practice, what does this look like? Well, in a normal control animal, what we would expect to see is that this baseline right here, where we injected glucose, is relatively normal. And then we have a large spike in the blood glucose levels. They peak at roughly 15 to 30 minutes, and quickly we renormalize blood glucose levels to normal glycemia. And that's within 2 hours, usually, that it goes back to normal. And the process by which this is thought to occur is called glucose-stimulated insulin secretion. So the bolus of glucose we administer leads to the secretion of insulin from pancreatic beta cells. This insulin then causes the uptake of glucose from the surrounding plasma into the organs, such as the muscle and the liver, and this leads to depletion of blood glucose levels and renormalization of normal glycemia. So, based on this, we would presume that if we were observing, say, hyperglycemia in these animals, they would be incapable of this glucose disposal mechanism. And in fact, that is what we observe in animals that we stimulate with our GQ drug. If we turn on these AVPOA neurons, we observe that not only within 120 minutes, which is the normal length of the test, but even within 240 minutes, which is twice the length of the test, we observe dramatic hyperglycemia. And to us, this reflects a glucose intolerance in these animals that is being induced by the activation of AVPOA neurons. Now, you might ask yourself, based on what I just said to you, well, could this all be explained by defects in insulin secretion? Perhaps the animals aren't secreting insulin, and this is the reason in which they're not uptaking glucose. So we performed ELISA assays in which we measured the blood insulin levels over the duration of this experiment. And we actually find that there's no statistically significant difference between control and GQ animals. And moreover, that the GQ animals have higher levels of insulin than their control counterparts. And so this is reflective of the fact that insulin secretion is not affected by the activation of these neurons, and that the explanatory factor must be something else. So with the knowledge that we systemically are altering glucose metabolism, as shown by that previous experiment, the question that then arose in my mind was, is this a general depression of glucose metabolism, or is this something where we can explain it via organ-specific defects in glucose uptake? And so to test this, what we did was the gold standard for assessing any sort of glucose or insulin insensitivity, and performed a hyperinsulinemic euglycemia. Now, the details of this are not tremendously important, but it is an experimental approach that allows us to not only understand systemic defects in insulin sensitivity, but also to address organ-specific changes in glucose uptake in response to insulin stimulation. And so we not only were able to confirm the insulin insensitivity that we previously described, but we use this approach to assess the organ-specific changes in glucose uptake. And so after performing this experiment, in which we used a radio-labeled tracer, C14-2-deoxyglucose, which is a non-metabolizable glucose analog, we sacrificed the animals and we took 6 organs. We profiled the brain, skeletal muscle, liver, heart, brown adipose, and white adipose tissue. And what we found was quite surprising. So whereas heart, liver, brain, and white adipose tissue show no changes, no statistically significant changes in the amount of glucose uptake upon stimulation of these neurons, we found that, in fact, brown adipose tissue and skeletal muscle exhibited 55 and 62% decrease in their glucose uptake upon stimulation of these neurons, these AVPOA neurons. Now, you might look at these rates and say, well, given how high the rate of brown adipose tissue uptake is, we might suppose that that's the dominant force behind the effect that we see systemically. But in fact, the skeletal muscle accounts for 65 to 70% of the overall mass of the animal, and generally is thought to involve about 80% of the postprandial glucose disposal. And so if we actually take these rates and we multiply them by the weights of the tissues, we find that the predominant driver of this phenotype is actually the skeletal muscle. And so we choose to focus on that going forward, because we believe that even though there is a decrease in brown adipose tissue related uptake, the predominant factor is skeletal muscle. So to quickly summarize what I showed in part 1, AVPOA neurons shift systemic metabolism, even in fed animals. These changes are organ-specific, temperature insensitive, and reversible. And the metabolic effect of these neurons is primarily a result of skeletal muscle and brown adipose tissue defects in glucose uptake. So now that we have a sense for what are the changes in metabolism that are actually occurring in these neurons, we wanted to ask the question, how is the signal conveyed from the brain to the periphery to actually affect these changes? And so that led us to consider 3 possibilities: temperature, direct innervation, circulating factors. Now, temperature we've already discussed a little bit here, but we need to understand whether specifically in the skeletal muscle, there's a temperature-related defect in blood glucose uptake that resulted from changes in the body temperature. Similarly, when you direct innervation, we know that there are 2 systems that innervate the skeletal muscle: sympathetic nervous system and the motor nervous system. And so we test whether either of those are contributing factors to the phenotype that we observe. And finally, circulating factors are known to regulate blood glucose metabolism, such as pancreatic hormones, adipokines, and adrenal hormones. So we first went out of our way to test the temperature effect of skeletal muscle glucose uptake and how ADPOA neurons regulate this. And so what we did was we combined a similar experimental approach to what we did in our RER experiments, where we elevated the ambient temperature. So we performed radioactive glucose uptake assays on animals at 24 degrees first, and assayed how the change in glucose uptake between the control and GQ animals looked like. And in fact, this is exactly what we would expect, in that there is a significant reduction in glucose uptake between the two. After this, we performed the same experiment, but with the animal's house at 32 degrees, and in fact observed, again, this dramatic reduction in glucose uptake as a result of GQ dread stimulation, suggesting to us that the glucose uptake defect we observe in skeletal muscle is insensitive to temperature. So we rule out temperature as a primary effector of this defect in glucose uptake metabolism, leading us to now consider the direct innervation hypothesis. Now, we first approached this from the perspective of the sympathetic nervous system. Sympathetic nervous system regulates many of the autonomic functions in your body. Think about fight or flight response, urination, pupil dilation, and so on and so forth. And the primary way in which it does this is by using the neurotransmitter epinephrine. So we reasoned that if we could perform what's known as a chemical sympathectomy or sympatholytic chemical intervention, we would be able to directly isolate the role of the sympathetic nervous system in this phenotype. And so to do this, we used a drug known as guanethidine. Now, what guanethidine does is it enters into the terminals of sympathetic nerves and displaces the norepinephrine from the synaptic vesicles. So that when you have released these synaptic vesicles, instead of releasing norepinephrine onto your tissue, you're releasing guanethidine, which is a nerve, and effectively gives you a chemical sympathectomy. So we first confirmed in our animals via glucose tolerance test that we saw the anticipated hyperglycemic phenotype. And then we injected them with guanethidine. And no surprise to us, we observe actually an increase in glucose tolerance when we just inject guanethidine. This is largely because we see vasodilatory effects of guanethidine administration, which lead to better perfusion of tissues and increased ability to uptake glucose. But the real question we had is, what happens when we do CNO treatment with guanethidine? And in fact, what we observe is we see almost no difference between CNO injected and the CNO guanethidine injected animals. In fact, no statistically significant difference, and we're unable to collapse the curve back down to control levels, which suggested to us the absence of sympathetic nervous system from our phenotype. So ruling that out, we then wanted to test the motor nervous system. Now, we used, in this case, a crude but effective experimental approach in which we wanted to target specifically the nerves that innervate the skeletal muscle. And so what we did was we performed surgical denervation by cutting the sciatic nerve. Sciatic nerve is the primary innervating nerve that goes to the lower limb skeletal muscles. And this allowed us to completely remove motor nerve input into these muscles. In the same animal, we were able to keep an internal control by leaving the other muscle innervated. And so this allowed us to do direct comparisons between control and GQ injected animals, comparing their denervated and innervated legs. So we performed our standard glucose uptake assay, and we found once more. We have our innervated legs, we observe a significant difference between the stimulation, the GQ dread stimulated animals and the control animals. When we looked at the denervated legs, we similarly observed this change, this alteration in glucose uptake into these tissues. And in fact, if you take the ratio of the denervated to innervated between the control and the GQ animals, we observe no significant difference between either the control or the GQ. And this suggests to us that though denervation results in overall lower glucose uptake in both the controls and the GQ animals, it does not explain the defects that we see between stimulated and unstimulated animals. So we rule out the motor nervous system. So finally, that leaves us with circulating factors. And we considered in this case 4 circulating factors. Considered the pancreatic hormones, insulin and glucagon, which have bodies of literature that explain their effect on blood glucose levels. And we saw no significant difference between either of these when we stimulated ADPOA neurons. Similarly, we looked at the adipocon leptin, which is the overall indicator of metabolic abundance of energy. We observed no difference in the leptin levels in these animals. However, when we injected them and observed corticosterone levels, which corticosterone is the mouse analog of the human cortisol, stress hormone, we observed a 2.59-fold increase in the levels of corticosterone. And so this led us to at least consider the possibility that corticosterone or some other adrenal hormone is involved in our phenotype. And so to test this, we wanted to ask the question whether or not adrenal hormones can actually mediate this phenotype. And so what we did was we performed another crude but effective experiment in which we performed a bilateral adrenalectomy, removes the adrenal glands, and effectively removes all adrenal hormones from the body. To validate this, we measured the levels of corticosterone in adrenalectomized or fam-adrenalectomized animals. We found that there was nearly complete ablation of corticosterone in these animals at the end of this experiment. Moreover, when we transduced these animals with GQ drugs and stimulated them with CNO, we found that the body temperature changes were identical, nearly, suggesting to us that any result that we got from this experiment would have to be independent of temperature if we saw a difference in glucose tolerance in these animals. So we took these bilaterally adrenalectomized animals and we went back to our standard glucose tolerance test. So in both control and adrenalectomized animals, we performed the glucose tolerance tests in the absence of CNO. And we found that there was no difference at baseline in between their normal glucose tolerance. However, when we injected these animals with CNO, we found a dramatic difference between the adrenalectomized animals and the sham controls. And that's quantified here, demonstrating to us that loss of adrenal glands actually is sufficient to completely ablate the phenotype that we observe with respect to glucose tolerance. So this put us into a paradigm, because we've done this more crude experiment where we've taken out the adrenal glands, in which we had to consider the two possible glucoregulatory hormones that could be acting here. So there are two sides of this axis. Hypothalamic pituitary or HPA axis, which is controlled by the trichohypothalamus, and the sympathoadrenal axis, which leads to the secretion of adrenaline and is controlled by the BLM. Now, we haven't shown the data, but we have reason to believe that we've ruled out the sympathoadrenal axis for two reasons. One, we don't see concomitant changes in plasma adrenaline levels when we stimulate these neurons, like serum. And two, this is controlled by the sympathetic nervous system. And so one would expect that we would have observed some defect in our guanethidine experiment if this were adrenaline would have been fired. And so for that reason, we chose to focus on corticosterone. And to do so, what we did was we used a drug known as meteropone. Meteropone is an inhibitor of corticosterone synthesis and effectively is a way to decrease corticosterone levels in the plasma, thereby removing it from the equation and letting us address what its effect is on our phenotype. So we performed these injections, or we treated these animals with meteropone. We found as compared to their baseline levels of corticosterone, we observed significant decreases in corticosterone levels, indicating that this was an effective way for reducing the levels of corticosterone in the body. We then took these animals and performed glucose tolerance tests. And I think you'll appreciate if you look at the peach colored line in the middle there, as compared to the purple one, we observed a flattening of this glucose tolerance test curve, indicating to us that removal of corticosterone from the system actually sensitized these animals to become more glucose tolerant, suggesting that the increase that we see in corticosterone upon injecting these animals with CNO is at least a partial, if not major, driver of this phenotype. And so, to conclude this section, saying these neurons induce glucose intolerance in skeletal muscle. This is affected by an insulin signaling pathway. The adrenal glands are required for the manifestation of this glucose intolerance effect, and corticosterone is the likely effector of this AVPOA neuron-induced glucose intolerance. So, for the last little bit here, I just wanted to return back to the normal physiology. Everything that I've described so far has been in the context of chemogenetically stimulated organisms. But we wanted to ask the question, what is the function of these neurons in normal physiology? And so we first came up with a model that would allow us to address this. And one could imagine something fairly simple, in which fasting these animals leads to decreases in blood glucose levels. This then leads to activation of these ABPOA V-glutathione neurons. And this leads to a decrease in muscle glucose metabolism, which then normalizes the decrease in blood glucose levels. And so this simple model, actually, we could test by using the torpor state as a natural physiological model to assess this. And so our first question was, we already know that these neurons are active during torpor. But if these neurons are actually responding to changes in, say, plasma glucose or nutrient availability, is their activity nutrient dependent? And so to ask this question, what we did was we transduced our animals with a genetically encoded calcium indicator, GCAM6, and we performed fiber photometry experiments by placing an optical fiber in the POA and observing changes in calcium signal, which is a proxy for neuronal activity in these animals over a 24-hour period. So we performed the following experiment to assess whether or not these are nutrient-sensitive neurons. We first fasted these animals for 24 hours and performed fiber photometry over that duration to confirm that these neurons were active during torpor, which we validated. And then after this, we refed these animals with either unpalatable clay or with a food pellet. And we monitored their activity for 30-minute intervals after the addition of either one of these things. And what we observed was quite interesting. When we applied the clay into the chamber, we observed no real change in the pre versus post levels of neuronal activity. But strikingly, what we observed is that when we apply food pellets there, we observe a dramatic reduction in neuronal activity. And so again, if we look at pre-addition, these levels are relatively normal. But after the addition of food, we see this dramatic drop in neuronal activity. And this to us indicates that these neurons are actually sensitive to the nutrient status of the animal, which is something that was not previously appreciated. So finally, what we wanted to do was actually test, can we affect blood glucose homeostasis by silencing these neurons during fasting? We know they're active during fasting, we know they're nutrient sensitive. Can we actually affect the phenotype that we've been looking at for the duration of this project? And so to do this, we approached it by two separate ways. We wanted to do acute silencing of these neurons, and we wanted to do chronic silencing. Now, to do acute silencing, we use what's called a GI dread. And you can think about this as basically the inverse of a GQ dread. It's sensitive to CNO, but instead of depolarizing the neuron and inducing neuronal activity, what we find is it hyperpolarizes the neuron, prevents neurotransmitter release, and actually allows for us to acutely and reversibly control the activation of these neurons by CNO administration. So we're acutely inhibiting them. The other approach that we used was this tetanus light chain toxin approach. Tetanus light chain is more of a chronic way to inhibit these neurons via the cleavage of this VAMP2 protein, which prevents synaptic vesicle fusion. And by doing this, it prevents neurotransmitter release and leads to chronic and irreversible silencing of these neurons. So we wanted to see what these animals looked like when we silenced these neurons. And so to validate the fact that we practically silenced them, we treated these animals with TLC, which is this tetanus light chain, and we observed that they did not go into torpor when treated with tetanus, as one would normally expect. So this indicates that our silencing paradigm actually was functional. And then when we considered blood glucose levels, we measured them from 0 to 6, 12, and 24 hours. And we found that at every time point in the animals transduced with tetanus light chain toxin, we observed a hypoglycemic effect, in that these animals had lower blood glucose than their counterparts that were transduced with tetanus light chain. And so this indicated to us that we could modulate blood glucose levels via the silencing of these neurons, but perhaps this chronic effect was actually leading to a change in the baseline. And so what we wanted to do was have a more acute approach. And this required the use of this GI silencing. So once again, we took these animals and we fasted them normally and injected them with CNO, or injected them with saline. And injecting them with saline, the animals go into torpor perfectly fine. The saline was administered at this 12-hour time point, right before the change between the dark and light spectrum. Now, when we administered CNO, we were able to block this decrease in body temperature, indicating once again that we were able to effectively silence these neurons. And so the question then becomes, what is the blood glucose level? And so we can see that at every time point before we perform our injection, the blood glucose levels are relatively the same between saline injected and CNO injected animals. But when we inject these animals with CNO, we observe that there is actually a hypoglycemic effect in animals injected with CNO as compared to their saline counterparts, indicating once again that these neurons are involved in normal glucose homeostasis. So I just want to make a final point here, which is to say, we see both in the overall condition here, so in the cell fasted animals, over the full course of the experiment, we see overall hypoglycemia. And in our acute condition, at the 24-hour time point, we see dramatic hypoglycemia when we silence these things. And when we looked at the literature to really assess how does this compare to regions of the brain that have actually been associated with regulating blood glucose levels, we find that there's really only one demonstration where people have tried this, and it's in one of the bona fide glucose regulating parts of the brain, ventricular hypothalamus. And in this study, when these authors silenced neurons in that region which are known to regulate blood glucose, they saw very minimal changes in the overall levels of blood glucose, significantly smaller than what we observed, indicating to us that our study is the first one in which we observe such significant changes in blood glucose homeostasis as a result of silencing these neurons. And so in my mind, that means that these neurons need to be put within the pantheon of neurons in the brain that are capable of regulating blood glucose homeostasis. So to summarize, we've shown that these neurons are nutrient sensitive, and we know that acute or chronic silencing of these neurons impairs normal glucose homeostasis. And as I said just a second ago, I believe that these neurons are bona fide glucoregulatory neurons, which need to be placed into the pantheon of CNS regions which can do this. So to conclude, say that we've shown that AVPOA neurons coordinate fuel usage during fasting, and they do this via changes in organ-specific glucose uptake, specifically in skeletal muscle adipose tissue. And they do this via engagement of the hypothalamic pituitary adrenal axis via corticosterone, which suppresses glucose uptake in an insulin-dependent and independent manner. Finally, these neurons are active during fasting and torpor, and their activity is regulated by nutrient status. And silencing of these neurons in contexts during which they're normally active dysregulates normal blood glucose homeostasis. So at the end, I just want to give a little bit of context on what we're doing for our future directions. And the first of which is we're going to try and determine the direct neuronal connections between these AVPOA neurons and their downstream partners that form the head node of the HPA axis, so these CRH expressing neurons in the paraventricular hypothalamus. We're also going to assess other contexts in which these neurons are active besides fasting and torpor. This could include stress response, heating, potentially other experimental conditions in which we can actually increase the neuronal activity of these AVPOA neurons. And then finally, in work I haven't shown, we've actually demonstrated that these neurons are fairly interconnected with a lot of downstream neuronal regions. And we don't have a great understanding of what these connections really do functionally. And so we'd like to first dissect the downstream neuronal targets of these neurons. And then finally, we want to figure out in the AVPOA, what are the minimal populations that regulate this glucose effect that we've observed? So with that, I'd like to thank my lab, especially the University team, who again, has been such a wonderful mentor to me and has provided me with the freedom to do this project and really take it whatever direction I want to. And has provided a tremendous amount of support with all of the co-op and technicians and visiting students who I've trained, including Matthew Alkire, Molly Madigan, Nate McGrain, Simon Opsal, and Audrey Wong, all of whom have been fantastic and helped tremendously with this project. Everybody else in the Hermitine lab has been a constant source of inspiration for me, and our collaborators at HMS, MIT, UMass, Koch Institute, and the Whitehead Metabolomics facility. All of you guys have been phenomenal, and yeah, really just appreciate the help. And so with that, I'll conclude briefly. And go to some acknowledgements first. I'd like to, just give a bit of a shout out to my family, who really have been, phenomenal supporters. Dad, Liz, Mom, all of you guys have just been, wonderfully supportive of my career and helped keep me on the straight and narrow throughout. all of that. And so really, really love you guys a lot, and feel fortunate to constantly, have your love and support. Other members of the family, John, Montija, and my Opa, all of whom have, been there throughout, my athletic and academic endeavors through most of my life, and really have, provided me with a base of support and new levels of happiness that I couldn't have predicted prior. And then the rest of the McFadden clan, Andy, Joe, Pat, Kathy, Steve, Deb, Laura, Claire, all of the Irish Catholic offspring who really form the crux and core of of a lot of my identity, and who have really been fundamental in my development. And so, yeah, a lot of thanks to you guys for all the help along the way. Friends, yeah, a lot of people to thank here. Rob, Julian, Kai, Carter, Huey, Mark, Leland, Tig, all of, many more that I'm forgetting. But all of you guys throughout the duration of my high school days, and even predating that, really have just been so great to have been with, and have really helped me disconnect from the science and sort of focus on things that actually matter in real life. And so that's been quite nice to do, and I just feel so fortunate to have you guys in my life. Friends from college, Vagilos program, the Clams clan, Ethan, Olivia, Sam, Henry, and Anne, both tremendous advocates scientifically, as well as, again, people who are able to keep me out of the lab for a little bit and thinking of things that aren't just scientifically related. And then the friends we made along the way, all of the people from my year who were so fundamental in helping me out, in those early classes, and, just being there to go for a drink or do something fun during the weekends. Running buddies, drinking buddies, Colin and Michael Stubna, just great guys all around, 2U Chicago boys, great guys. And yeah. This brings me back to Sinisha. And, Sinisha's a man of many faces, many colors, many characters. But I have three representative forms of Sinisha, which I'd like to demonstrate here. And the first of which is lab mode, where he's sort of, this erudite scholar who's kind of locked in and ready to address all of the greater questions that the scientific literature demands. There's that mode where, he is he's a father of 2 kids, and actually really is an inspiration for showing that you can be, a parent, but also extremely successful at a high octane research program, and be really good at your science. And then there's party mode. And this one is interesting when it comes out. He's never afraid to throw down on the dance floor. This is something that I found out early in my tenure in the lab. Have been consistently impressed with his desire to go out there and just let loose. Very humbling to see those dance moves. So, but in all seriousness, yeah, Sinisha, again, he's really been a huge advocate for me throughout my career, and really in a time when I wasn't, things weren't going so well, when I was transitioning labs. He was there to pick me up in ways that, not everyone was willing to do. And he took a chance on, a 3rd-year, slightly radioactive grad student. And, I hope that I've repaid that confidence and that trust, and been able to be a productive member of the lab. So really just appreciate your help with everything scientifically throughout my career. My committee. So despite the fact that I try and avoid committee meetings, these guys have wrangled me into a few of them. And in all of them, they really do have this way of pushing me to think about the problems in ways that I necessarily wouldn't. I sort of fall at this crux between metabolism and neuroscience, and so I get a lot of good feedback from people who are either a real neuroscientist or a real metabolic biologist. So it's refreshing to, again, despite the few number of committee meetings, really have their support throughout the duration. So thank you guys. And thank you again to Shingo for coming out today. Really appreciated taking the time to come across the bridge, all of the circumstances, be damned. and make it here today. So thanks again for coming out. I'd be remiss if I didn't bring up my previous PI. So as many of you will know, I didn't start out in Sinisha's lab. I started out in David Sabatini's lab. And, you know, it was sort of a foundational scientific experience for me for my graduate career. And I just can't say enough about how David created a great culture for his lab that really pushed you to be your best and really helped me meet some truly fantastic scientists and people along the way. The lab, we'll get through these, I promise, but the lab has been, you know, absolutely phenomenal place to work. We, as you can tell, we're a very social lab. We do our own retreats, we go to other retreats, we bowl, we eat, you know, we cook on Cat's roof. Yeah, we do a lot of things together. I'm not necessarily the best person at making it out to these things, but they really are, you know, really fun people to hang out with. Who also in the lab are, you know, fundamentally able to push me and inspire me every day. And so I have a lot of love for you guys and a lot of thanks for you doing all of that. The friends I made along the way in the Sabatini lab, we've got a lot of people here, you know, Genya, Jordan, Jabril, Paul, Anna, Jess Spinelli, the whole lab, the whole gang. And these two guys here who I really know, Paul Rosen and Hank Adelman, really just two good guys, two good dudes, who have helped me not only as a person, you know, but as a scientist. Really pushed me through, you know, a little bit of bullying, a little bit of incisive commentary, and gotten me to the point that I am today. So a lot of thanks for you guys. Finally, a shout out to the Berger Lab. This is where I did my undergraduate work, and Shelley Berger, Carl, and Yanko all kind of, you know, put me on this path today. So if you want to blame them for that, you can. If you want to thank them for that, you can. These are their names and addresses and such. But you know, they were tremendously supportive of me in my early scientific career, and so I really appreciate what they've been able to do for me. And then finally, my partner in crime, partner in life, Tiffany. You know, she's been there for me for quite a while, and we do everything together. And she's really there to pick me up when things aren't going so well in the lab, and I just need to recenter myself. She's been a rock in that way, and so really a lot of love for her, and a lot of feel very thankful to have her in my life. And then finally, the last one. I know we don't like to think about this, but you know, this project would not have been possible without the sacrifice of quite a lot of mice. And so I do want to just acknowledge that sacrifice, and that, you know, we can't do this type of biological research with animals without these little guys. So a moment of silence for these little dudes. Okay, and so that on that morbid note, thank you all for coming out, and would appreciate any questions and comments that you guys have.
Hey, awesome talk. I have two questions.
Yes.
Sort of related. So I guess you said that you didn't want to focus on brown adipose tissue, but I'm curious if the inhibition of corticosteroid also rescued those phenotype in the brown adipose.
Yeah, so we haven't tested that at the level of uptake, actually. It's something that we're planning on doing, but we really have just chosen to focus more on the skeletal muscle, just because we think it's the dominant factor there. We haven't done that sort of deep dive into the direct mechanisms within the brown adipose, and it's not to say that they do have to be identical to what's happening in the skeletal muscle, because there are known projections that go from the preoptic area that transsynaptically proceed directly to the brown adipose tissue. And these aren't appreciated in the skeletal muscle. There's actually not a known pathway from the POA that directly projects the skeletal muscle, but there is one for the brown adipose tissue. And so you could imagine that there'd actually be different modalities by which this signal is conveyed to different organs. So still an open question, but something we'd like to ask.
Great. And my second question is just, like, what do you think mechanistically is driving the tissue specificity of this in those uptake phenotype? Like,
Yeah.
Is it just regulated through blood or?
Yeah, so the main answers we have from that, again, we focused on skeletal muscle, and what we've done is some biochemical analyses. And we've seen that, yeah, defects in the insulin signaling cascade, which would lead to GLUT4 translocation, are actually they're hypophosphorylated and therefore reduced in skeletal muscle in response to stimulation of these neurons. And so we do think that that may be part of the reason that you have this specificity, because it's known that at the very least glucocorticoids they have pleiotropic effects in different organs, but specifically with respect to skeletal muscle, they're known to impact glucose uptake directly there. And it's possible that it could be going through just impairment of insulin signal in general.
Questions also. One, is there any evidence that these pathways are active in humans, maybe with a stroke patient?
Yeah, so I would say we don't even have any direct evidence that humans necessarily have, these types of B-glue neurons that are active within the preoptic area. My general sense, based on some work that Chris has done, some work in more sense, suggests that the neurons probably do exist there, but whether or not they're wired in the same way, we don't really have a sense of that. And that would be ultimately the determining factor in whether or not they would perceive biosimilar pathways. Is, are they actually wired similarly? So we don't know.
Other questions, given how exciting this magnified. Can you speak on whether these neurons have
Genuinely, I don't know. I'm sure Chris has an idea right now. But no, I actually don't know. They do have, there are known like insulin related receptors that are present within the preoptic area, but I don't know specifically what the GLP-1 is specific. Okay.
Is it known if there's a critical period for maturation of POA, and is there a period when I said the first 21 days, where you can have slower responses, and requires some period of which?
Yeah. So it's actually very interesting. So the mice don't really thermoregulate that well for roughly the first 10, 14 days of development. They basically require exogenous heat from their mothers or their other litter mates in order to maintain body temperature. And so there is clearly a window in which this wiring has to occur at the very least. Now, once you make it past that 14-day window and you're actually like a weanling that's ready to go out into the world, possibly there are differences between that and an adult. We've actually never tested like a weanling, if you fast them and have a probe in them, whether or not they go into torpor. My sense is that they probably would, though, once they have a fully developed regulatory system. That just would be most of my finding.
Great. So let's thank again Julian, and then we'll see some of you in some of the time in about an hour or so.
Okay. Thank you. Oh. All right, all. We are out. Thanks for coming out, guys.